you doing? It's been a while.
Yeah, since the beginning of the first lockdown, right?
Yeah, what have you been up to?
Oh, not so much, just working on iGEM.
Oh, so you're not applying for the team?
Yeah.
What was your project again?
At the beginning of the year, we got the opportunity to attend a presentation given by the 2019 MSP-Maastricht iGEM team about what synthetic biology is and what the iGEM competition aims to achieve. In a room full of curious students, we were all excited to create our application to join the team. We had all heard about iGEM and we were eager to get involved. We sent in our application on what we contribute to the team and an idea of a problem to tackle during the competition. After a few weeks of waiting in full excitement, we all received our confirmation mail about being part of the official MSP-Maastricht iGEM team. The team consists of 11 MSP bachelor students, 1 bachelor student of biomedical science, and 1 master student of system biology. On top of that, we were guided by 2 incredible supervisors, Eric and Ben. It was finally time to meet the rest of the team members. Just one problem: Corona. This meant we could not meet in person. Together with the new team, we started the journey of finding the right topic. No one is safe if it is for the oak processionary caterpillar, as the invasive species affects fauna as well as the flora they reside in. They strip trees bare to their bark, making the oaks vulnerable to harsh environmental conditions. For animals and humans, the OPC poses a serious health risk as a result of their toxic bristles. Rashes, severe itching, eye complaints, and respiratory issues like asthma are common symptoms evoked by its allergenic protein, which triggers inflammatory responses. In the most serious cases, bristles even have to be removed surgically from the eyes. As a consequence of the soft winters and drought that climate change brings along, the OPC returns with vengeance yearly. The global phenomenon seems to be the OPC's best friend, as it allowed the species to expand its territory from its native region in southern Europe towards many European countries, and even parts of the Middle East. Over the years, the invasive species has become a major local problem in the Netherlands, since more than 100,000 people were estimated to be affected by this caterpillar in 2019. Municipalities annually have a hard time managing the problem, and this is how we came in contact with Jules Sondijker, who coordinates the control of the OPC in the Dutch province Limburg and is connected to the municipality of Sittard-Geleen. As an expert in his field, we were in close contact with him, as he has guided us in shaping the project with regards to biosafety and efficacy of the product. Also, the knowledge we gained from him in interviews about current measures were indispensable in making our new product stand out from the rest and determine the necessary features to develop an insecticide that combats the OPC in the most environmentally friendly way. These current control measures are non-specific, meaning they are dangerous for the caterpillar as well as other species that reside in the same niche. Consequent to this, biodiversity in the area is highly affected, enabling the invasive caterpillar to grow in population due to a lack of natural predators. This will result in an imbalance in the population, causing a pest.
Amazing idea. How is it going? Are you done with the wet lab?
As a first step, we identified essential genes of the OPC. The silencing of these critical genes with matching silencing RNAs is expected to lead to a decreased expression of the target genes, cellular senescence, and eventually to the death of the caterpillar. We identified 4 potential target genes: the allergenic protein TARP2 gene, the Pro2 photolyase gene, which is essential for proline metabolism, the wiki gene coding for wingless protein, which is thought to be involved in numerous developmental processes, as well as the EF1 alpha gene for elongation factor 1 alpha, which plays an important role in the elongation phase of protein synthesis. The selection of specific silencing RNAs was done by selecting 21 nucleotide sequences that start with 2 adenines and running them through the BLAST algorithm to identify sequences that are not found in any other organism, thus ensuring specificity only towards the OPC. While we were selecting targets for our silencing RNA, we came across many different possible target genes. Unfortunately, due to the global pandemic going on, we had no access to the lab and no way to validate our targets, so we started looking at possible models to use to determine lethality. We eventually settled on metabolic model built using analogous RNA expression data linked to a mammalian framework because no arthropod framework existed. This allowed us to test and validate all but one of our possible targets regarding the detrimental effects when silencing them. With the help of this model, we were able to change our construct if needed before starting wet lab work and thus conserving and saving already limited resources and time. The dry lab team additionally created a mathematical model for the spread of the OPC population in an area. This model is based on the spruce budworm model by D. Ludwig, a dynamical system model with multiple steady states. When the sections for population growth and predation in the formula are equal, the population is stable. With this model, we aimed at gaining a better understanding about sudden OPC population changes leading to outbreaks. After working on the dry lab research, where we found the target genes we wanted to test, the experiments could begin. The first step was to find these target genes within the OPC genome and amplify them. We extracted the OPC DNA and quantified it. The team then proceeded to run multiple PCR tests using the specific primers found during dry lab research, which would amplify the sequences of interest. Once the fragments of interest were amplified, we ran them through gel electrophoresis to see how successful the PCR was. The promising bands, TAPI2, wiki, EF1 alpha and Pro2, were extracted from the gel. During this, we prepared a Pro2 sample from the OPC to be sent off for sequencing. The Pro2 sample came back as 100% identical. Later on, we sent off 7 more samples for sequencing, including wiki, EF1 alpha and TAPI2. The RNase-free strain of E. coli bacteria was cultivated and grown in petri dishes overnight to be used in oak tree testing, meaning the bacteria were sprayed onto the leaves of gathered oak branches and tested for their presence of 24 hours by plating them on ampicillin selection plates. This was done to determine how long the bacteria could effectively stay on oak trees. The plasmids were linearized with restrictions enzymes, and the shRNA coding DNA fragments were ligated into the plasmid. The full plasmid then contains the silencing RNA in an expression cassette and is transfected into the competent E. coli cells. These cells were grown and plated on ampicillin selection plates and incubated overnight. Their plasmids were extracted and transfected into the selected feeding strain of E. coli. Finally, we induced the production of silencing RNAs with IPTG and checked for their presence through gel electrophoresis.
Unfortunately, our final E. coli strain did not seem to produce shRNAs after IPTG induction, as there was no specific band during electrophoresis. To confirm that our siRNA inserts were assembled into our vector, we performed a restriction digest with ECHR1 and BAMH1 to remove and detect our siRNA inserts. However, the predicted band did not show up on our gel, indicating that the siRNA insertion step did not work. Nevertheless, we will sequence the produced plasmids to confirm this, and if it was indeed not successful, we will repeat the step.
That's nice. I also saw you making videos on YouTube.
Oh yes!
I don't remember how exactly we came up with the idea of having a YouTube channel, but very quickly, in April already, it was an integral section of our project. Where a part of the team would be leaders in the dry lab work, others were focusing on our channel. Our intention was to, in spite of the lockdown restrictions, reinvest our time in making educational videos about synthetic biology and genetic engineering. We came up with the name, Gene Education, and started to write scripts. We knew that the bad reputation of GMOs in the public is often mainly due to lack of information.
Well, typically when you say your non-scientific friends or family you are making use of genetic modification in a project, then you see disapprobation in their look. But if you say, I'm using bacteria to degrade plastic in a sustainable way, they are quite interested and enthusiastic.
That's why we decided we would start from scratch in the popularization of cell molecular biology. We created together a whole list of scripts going step by step into our genome editing and GMO use. This stepwise approach was necessary because we included people with little to no scientific background in our target audience. Then we had to create an opportunity for some of us to meet and do the shooting with professional equipment. At a time where many countries had closed their borders, this wasn't an easy task. Eventually, some of us managed to and had to face the camera. We then had to learn how to edit educational videos, as none of us really had experience in this field.
The CRISPR-Cas technology, which is short for clustered, regularly interspersed palindromic repeats.
Finally, on the 10th of May, we published our first video, introducing iGEM and cells as the smallest unit components of living organisms. This one has now more than 400 views. Then the months went on and we published more and more videos, also including do-it-yourself home experiments like DNA extraction or enzyme assessment. We also started collaboration videos where we would present other team projects. For example, we took advantage of the first Aachen meetup to record one video together about ATP and their Mars project. Because we wanted to reach the broadest audience possible, we also tried to include subtitles in many various languages, also by collaborating with Team iBov China. We also created special content for kids and used this platform to share our feelings about iGEM in pandemic conditions by self-interviewing each other with the teams from Aachen and Nantes. In total, we now have posted about 20 videos, and we have 80 subscribers that regularly share their feedback on our work.
Wow, that sounds like a lot of work. I can see you didn't waste your time.
Wait, and that's only the beginning.
A big part of our iGEM project are the human practices and our journal initiative. From the very beginning of our iGEM journey, we had the idea to create a proceedings journal that would include research papers from all iGEM teams. We worked hard to make our idea into reality, and in the end, we had managed to collect 43 articles from 32 teams that were willing to participate. The way our journal worked was exactly like any other scientific journal, involving peer reviews, meaning that any team that submitted an article also peer-reviewed other teams' articles. This was done through an open review system designed by us to create a fair peer-reviewing process. Our team decided to create not only an online version where all articles submitted can be found, but also a printed version where the best papers rated by all the other teams can be found. We also know that articles in scientific journals are not read by people without the necessary scientific background. For this reason, our team created a Muggle journal that includes similar content as our proceedings journal, but in a much more understandable language for individuals with less scientific knowledge. With this, we want to make scientific articles accessible for everyone.
That sounds great. Did you ask people what they think about your project?
We made a survey to get more knowledge on what the people think about the OPC problem in general, the current methods to fight this issue, and our approach with Oak Shield. We especially wanted to know what people think about using synthetic pesticides and GMOs to fight the OPC. In the end, we learned a lot from this survey. First, the survey gave us much info on how people view the OPC problem. The majority of people from the countries that are affected by the OPC know what it is and either have encountered it personally or heard of people else encountering it. We also asked about the control methods. It turns out that on average, people think that our method would be more effective to control the OPC compared to current methods. Another key finding was that even though people are in general cautious towards using synthetic pesticides and GMOs, using these methods to fight the OPC made the participants view these methods more positively. Because we also included a question about the media coverage of the OPC, we learned that radio is an easily accessible media outlet that really lacks coverage of the OPC. This motivated us to create a radio show presenting the OPC and our project on RTV Maastricht. We also made a survey for our YouTube channel, which was more a small-scale survey, more like a study. We got people to watch a few of our videos, and then we asked them to answer questions on both the quality and the content. We got overall really positive feedback in all aspects of our videos, but we also got really good suggestions and comments on how to further improve what we are already doing.
Have you already checked out our latest videos?
Yes, I also love the collabs you did with the other teams. There were quite a few, right?
Throughout our iGEM journey, we invited other iGEM teams to make videos with us. Besides our educational YouTube channel, Gene Education, we had other great similar collaborations, ranging from lectures and podcasts to panel discussions and workshops. One example is the presentation of our project in a panel hosted by the iGEM team from Leiden, where we discussed how our project provides a solution to a local problem. With the team iBout China, we created a coloring book for kids and recorded podcast episodes discussing synthetic biology. We also have a long list of things that we have done by ourselves to spread awareness about the OPC problem, to educate people on our approach and promote iGEM and synthetic biology in general. One way in which we accomplish this is by writing blogs about our iGEM journey on the website biotechnology.nl, a prestigious Dutch website that translates today's scientific research on biotechnological topics to students of high school level. Additionally, we presented the local issue we are trying to resolve and the iGEM competition to the local population and our university peers to educate them on what is going on in their near environment.
I remember that one. You're really convincing me to apply for the iGEM team next year. But other than that, how are you doing?
Quite nice, actually. I'm finally trying to get some time for my own now.
Hey, we need to make
Hi.
a 20-minute video presenting what we did for iGEM for tomorrow.
What?